So now we go to the processing and quality control of single-cell RNA-seq. So depending on whether you do, so we're only talking about SMART-seq and DROP-seq for the course. Depending on whether it's SMART-seq or DROP-seq, you know, nowadays people are sequencing much longer. For SMART-seq, you do paired-end sequencing, and on the left and right, both are transcript. There is a barcode at the end because you do, basically each cell you make a library, and right before you put them for sequencing, you add a little bit of a barcode just to say, this is my cell 1, this is my cell 2. This is no difference from multiplexing 10 or 50 RNA samples into the same sequencing lane. They do that, so there are some, you can use standard RNA-seq method to separate out the barcode afterwards. But with droplet, it's like this. In one end, you do see the transcript sequence. You know, you do PE150 or PE100 base pair. One end of the read is mostly the sequencing. The other end, you can see here, there is a cell barcode. There is also a UMI. So the UMI is kind of some random sequence that you add at the end of the transcript. Therefore, if it's coming from a different transcript, initial RNA, it will have a different UMI. And then remember at the end of the transcript, there are always a lot of poly A. And so initially to do the alignments, you did the poly T. And so basically every cell will have a different barcode, and then every transcript will have a different UMI. And so there are some pre-processing, you know, demultiplexing, and also figuring out, you know, the barcode and the UMI from each of the paired end read. And then, so there, after the poly T, you might have a very short sequence left. Depends, you know, I think nowadays when people are sequencing 150, they might have 50 base pairs left after the poly T to be still able to map to the read. But then the other end, so here we say it has a 3' bias. It does not mean that it's the sequence right before the poly T, because, you know, when you have a paired end sequencing, poly T is here, and remember that if you run RNA-seq, the insert between your reads are usually, you know, 1 to 200 base pair. So it's towards the later half of the transcript, but it could still be a few hundred bases there, right? So it's not just at the very end of the transcript. It could be a few hundred bases towards the end of the transcript. So yeah, so you can see there with droplet, one read is mostly focused on the cell barcode, UMI, and the, you know, poly A on the transcript, which is poly T on your primer. And the other read is really the cDNA. And then you can map this into the genome. You can do the standard RNA sequencing mapping, which is STAR mapping. So the STAR group, they recently also developed a tool called STAR Solo, which can deal with all the barcode UMI automatically, which you can use. You can also use pseudo aligners such as Callisto and Salmon, which only map the read into the transcript, because, you know, most people doing single cell are not really even looking at new genes or novel transcript or isoforms. They just need a readout for the gene. Okay, so you can use pseudo aligners as well. And then the important thing is, after you read map the reads into the genome, we need to create this expression index. By the way, if you are using 10x Genomics, they already have a tool called Cell Ranger. So the company now also have computational biologists who are developing some basic tools. You can use their tool, or you can use STAR Solo to also generate this single cell matrix. For the homework that we are going to give you for homework 3, we will already give you this matrix. And so basically what you will see is that in this, it's a big matrix. Every cell is one column and every gene is one row. There will be a lot of zeros in here because of the dropout. Remember, each single cell from a 10x genomics, you only read out a couple thousand genes probably. And so the remaining genes are all gonna be 0, right? And maybe in another cell, there'll be other like 2,000 that has numbers and the rest are 0. And in the count here, that's important. Every time you see a read that's mapped there, if it has the same UMI, you collapse all of them into one, because you know that this is just PCR amplification bias. They came from the original same transcript. If it's mapped to the same gene, but they also have different UMI, that's when you add the count. You only count how many unique UMIs have read that are mapped to this gene. And that's what is in this table, okay? Because if it is the same UMI and the same cell barcode, that means it's the same RNA, and you're just PCR amplifying probably too many times. That's why you are reading it over and over again. Okay, so you compress all the transcript within the cell barcode, and if they share the same UMI, you count them as just one. And so anytime you see this 24 or 41 is because for that gene in that cell, you see 24 different UMIs of RNA mapped there. So interestingly, for SMART-based approach, they don't use UMI. The amplification method is more efficient. You just get the read count. For droplet-based, because they are dealing with so many cells, the reagent, they have to make sure everything can amplify. So they amplify a lot, and UMI is for a way to control for the PCR amplification bias. And after you have this cell count matrix, you can also convert this into a TPM or FPKM, similarly to what we did with RNA-seq. Just one thing is, for droplet-based data, because you are dealing with mostly the 3' end of the transcript, you may or may not want to normalize by gene length, because it doesn't matter how long this gene is. You're only able to capture the end, right? So you just do. So the normalization is, I think, easier. Just how many reads do you see in this total cell, and so on. Okay. So after we have the cell read mapping and also the cell expression matrix, we need to do some basic quality controls on the cells. And this is related to underloading or overloading of your the 10x genomics machine or whatever single cell droplet-based machines. If you underload your cell, it would mean that the reagent droplet is still going in, but you are not giving it fast enough cell coming in, which means there will be a lot of droplets that are empty. That's not so good. So after you waste all your reagent, you only get 2,000 cells instead of 8,000. But if you overload the cell, there will be a lot of cells, a lot of droplets that have multiple cells in there. Then it's also problematic. And so in the early days, when people were trying to just calibrate their machine to see how fast can I feed the cells into the machine, they do something like this. They get some tissues or single cells from mouse, and they also get some tissues from human, and they mix the cells together to run this machine. So you can see this is how many transcripts do you get. So this is without considering so how many unique UMIs in some sense, right? And after mapping, remember, if you map the RNA of human, 99% it knows how to map. Or even though human and mouse have very similar coding sequence, very often you can know whether the transcript is coming from human or a mouse. If you load the machines at, say, 50 cells per microliter, you can see there will be some cells that have transcript coming from both human and mouse. And you know that this is probably a little overloaded. Whereas if you load it very lightly, you can see there's very few of this mixing. I would say even at this level of mixing is not as bad. You cannot avoid those doublet. Also, there are cases when you can see, some of the transcript, some of the cells you see many transcript, whereas other cells you see very little. Very little could be because maybe that droplet didn't have enough reagent, or maybe that cell has died and the RNA is starting to degrade. And so various problems. And so usually there are algorithms now in the homework, you will use them. They do some standard quality control. For example, if with one droplet, you see this cell barcode, but a very, very overall low read count. This is usually, it's just not amplified efficient. Library preparation failed or whatever, or very low percentage of mapped reads, or very high percentage of mitochondria reads. This usually indicate the cell has died and there are only mitochondrial RNA left. What we will be using mostly is if you detect too few genes in one cell or too many genes in one cell, because this experiment is kind of a calibration experiment. Once you do your samples, you never put human mouse samples in the same run, right? You always do it just your own, say, a bunch of tumors going through treatment or whatever. So you can't really tell whether it's a mixing, like in this case. Therefore, the computational algorithms that we will be using, they show you overall a distribution of how many genes you are able to detect. And so you can decide a cutoff to say any cell that has maybe less than 500 genes, I'm gonna not consider them. And then you can see if the mean is here, then some cells have so many transcripts detected, it's possible because two cells have gone into the same droplet. And so you might also eliminate that. Say for a 10x genomics, usually you detect 2,000 cells, and then in this one cell you detected 4,000 genes. That's like not very likely. So you can eliminate those. Okay. And yeah, also some of them might lack a cell marker gene, and you can also see, maybe the intergenic intron exon read rate, and whether there's some bias. But I would say most people just use this very quick way to filter genes. So you can see initially with your cell barcode, you might end up sequencing 9,000 cell barcodes, but after this read mapping and QC, you might end up with only 6,000 left that has a reasonable number of genes detected per cell. Questions about read mapping and quality control?